the hypothalamo-pituitary axis. We spoke about the trophic hormones that are released from the hypothalamus. We call them primary trophic hormones. And then there were some secondary trophic hormones. We drew out that entire axis on Friday and just showing how the hierarchy of the endocrine system really works. So we've got the hypothalamus. I've been describing hypothalamus as kind of the CEO, the gland that's in charge of everything, so to speak. Then the second in command would be like the pituitary, the anterior pituitary specifically, releasing those secondary hormones. And then the third would be all of the endocrine glands, and then they also have their secretions, okay? We also spoke about the posterior pituitary. We don't want to confuse the posterior pituitary. It's a different type of tissue. It's neural tissue as compared to the epithelial tissue of the anterior pituitary. And then the hormones that are released from the posterior pituitary are not actually made there. We talked about that, that connection of neurons that are coming from the paraventricular, from the supraoptic nuclei that reside in the hypothalamus that kind of let those hormones down where they're stored and released from posterior pituitary. We got here to speaking about the regulation of all of this. So how do we determine how much hormone is released, when hormone secretion is too high or too low? And there are certain inputs here that will control that will regulate that. So we've got neural input. We know about these neurons, afferent neurons that are coming up to the central nervous system. They're bringing information in that can influence the extent and the amount of hormone release from the hypothalamus and pituitary. We've also got this negative feedback, which is hormonal. So the hormones that are secreted from the endocrine glands themselves, we think about thyroid hormone, we think about prolactin, we think about cortisol. Those hormones can then have a negative feedback mechanism onto the hypothalamus and decrease the initial primary trophic hormones, the amount of that that's secreted. So that's a second way that we can regulate this. The third method of regulation here has to do with the circadian rhythm. And this is a very small percentage of the endocrine glands, but some of the glands, specifically the pineal gland, is sensitive to light, to our light and dark cycle or our sleep and wake cycle. And that's what the circadian rhythm is. So for example, the suprachiasmatic nucleus of the hypothalamus, another cluster of cells in the hypothalamus, is sensitive to light. So when we're up and awake in the daytime, lots of light coming into the retina, we decrease melatonin secretion, and that is what keeps us, aroused and focused on our daily tasks. During night, when there's no light coming into the retina, the suprachiasmatic nucleus will then instruct the pineal gland to secrete melatonin, and that will make us restful, sleepy, drowsy, and go to sleep. So the circadian rhythm, the amount of light and dark that's being exposed to the retina, also has a regulating influence on hormone secretion. Let's look at this flow chart here. So this is just an example of this axis and the negative feedback loops. We've got two types of feedback loops. We've got a short negative feedback loop, which is basically the anterior pituitary, the secondary trophic hormone, having a negative feedback onto the hypothalamus. And then we can have a long feedback loop where the endocrine gland or the tertiary hormone that's secreted as a part of this axis will then feed back onto the hypothalamus. So both of these will then go back to the hypothalamus and decrease the initial amount of that releasing hormone that's being secreted, basically slowing down the upstream event of this hormone secretion. And then lastly here, we'll look at the pineal gland. So we talked a little bit about it just now. It's in the brain. It's kind of posterior to the thalamus here. It's responsible for secreting melatonin, which we know is involved in our sleep and wake cycle. It's also important in other seasonal processes in mammals. So for example, the ovulatory cycle, the menstrual cycle in women. And so again, this is thought to be controlled by the amount of daylight, sunlight, and the absence of daylight that we're exposed to that's coming into the retina, and other seasonal events that have physiologic consequences. Okay. So that brings us to the end of packet 1, which really just introduced the glands, really talked about identifying the primary from secondary glands, and then talked about the overall hypothalamo-pituitary axis. Let's move on to the second packet here. Actually, stop this. Okay, so let's kind of switch gears, move on to the second pocket, and talk about some specifics as far as some of the hormones are involved. We'll also talk about energy metabolism a little bit later on. So here we start out with this fourth objective, which is speaking about the synthesis and secretion of thyroid hormones, and then distinguishing between direct and permissive actions of thyroid hormones. So the thyroid gland, one of our endocrine glands, it's anterior to the trachea. We can see it here. It covers the first couple of tracheal rings here. It also has several lobes, so 4 lobes, as well as the parathyroids embedded in the posterior aspect of the gland. So we'll talk about the parathyroids a little bit later on, but they're really kind of stuck in the back part of the thyroid as well. The thyroid gland secretes 2 important hormones, T4, which is tetraiodothyronine or thyroxine. This is more involved in the feedback, and I'll talk about that. There's also T3, which is triiodothyronine, and this is more involved in the actual metabolic role of thyroid hormone. So here we can see that the role of thyroid hormone is really regulating metabolism. And when we speak about metabolism, this is just the widespread rate of energy use throughout the body. And we'll talk about some of the exact examples of how metabolism can be sped up or slowed down, but thyroid hormone pretty much governs our metabolic rate. There's also calcitonin that's secreted here as well. So there are some other cells, and I'll point the different cells out. So there are different cells located in different parts of the thyroid gland. Some of them involved in the thyroid production, that is T4, T3. Other cells, and I'll point out the difference again, are involved in calcitonin production. And the role of calcitonin is to regulate calcium levels in the blood, specifically to decrease calcium levels when they're too high. Okay. And I'll talk about an antagonistic hormone to calcitonin again a little bit later on. So just some really important notes to make about T3, T4, calcitonin, and where they're secreted throughout the gland. So let's start by looking at thyroid hormone specifically. It's released from these follicles. So within the thyroid gland, histologically, we can appreciate these subunits, these functional subunits called follicles. This is where the thyroid is made and stored and released. And so if we look at a follicle, it's lined by these cells. These are called follicular cells, making this a follicular epithelium. Inside the cells, we've got this colloid, and colloid is really the storage form of what becomes the thyroid hormone. And that's interesting because if we look at things like a goiter is a swelling of the neck caused by a decrease in the amount of thyroid hormone released. This is what's actually building up and swelling up, that's creating that swelling in the neck that's associated with a goiter. So this is really the storage form of what should become the thyroid hormone. It's composed of thyroglobulin, which is a precursor for the thyroid hormone, and that's pretty much located throughout the colloid. Now, in order to actually release this thyroid hormone into the blood, we've got to have iodide present, which is the ionized form of iodine, right, taken in through our diet. This is why that's so important to be supplemented in the diet. And that is used to join with the thyroglobulin, which is the precursor, and then that makes thyroid hormone, either T3 or T4. That can be released from the thyroid gland into the bloodstream, and it can make its way out to the peripheral tissues where it will have an effect on the metabolic rate. Okay, let's talk about the mechanism of how this actually happens. So here we can see this is the inner surface of the colloid, or the inner surface of the follicle, I should say, where we have the colloid present. This is the follicular cell, or the follicular epithelium lining the follicle. And then on the outer surface, or the apical surface of this cell, we can see exposure to the blood vessels, okay? Let's talk about what happens to the thyroglobulin. So colloid is pretty much made of thyroglobulin. Iodide has to come in here. So this is where iodide comes in. It's going to create these residues, either diiodotyrosine or monoiodotyrosine, depending on how many of those iodinated residues are combined. We get either either 2, which is diiodotyrosine, or 1, which is monoiodotyrosine. And we basically combine those with thyroglobulin to create either T3 or T4. T3 if we have a diiodotyrosine and a mono, which is 3 residues, or T4 if we have 2 diiodotyrosines, which are going to be 4 residues, okay? So that's basically the structural difference between T3 and T4. Otherwise, they're pretty much the same, bind to the same receptors, bring about the same effects for the most part. That can be taken into the cell via endocytosis, and that will then be transferred to the bloodstream across the cell. Let's talk about what has to happen on the cell membrane first before this can be uptaken into the cell. So we've got TSH. Remember, TSH is one of our trophic hormones coming from the anterior pituitary. If you kind of back up here a little bit, it's going to be released in response to TRH coming from the hypothalamus. And then once TSH is released from anterior pituitary, makes its way down to one of these follicular cells, binds onto this TSH receptor. This is a metabotropic receptor, so a great example of how this slow receptor works. It is coupled to a G protein. We've talked about these events, and that brings about this production of our second messenger, cyclic AMP here. Protein kinase can phosphorylate a protein in the cell, which results in this endocytosis of T3, T4. Okay, so this is an example of that second messenger pathway that we've talked about for other receptors as well. So that T3, T4 gets engulfed into the cell. It travels across the cell membrane, eventually leaves the cell via exocytosis, goes right through the membrane. We'll talk about the structure of thyroid hormone, but it's lipophilic. And so it doesn't need a carrier or a transporter or anything. It can go right through that, that phospholipid bilayer and make its way into the bloodstream. Okay, and that is how we go from iodine and the thyroglobulin that's stored here in colloid to actual T3, T4 entering the blood. So let's talk about the functional aspects of thyroid hormone. So it's lipophilic, we said. It acts like a steroid, it behaves well with fats, goes right through the lipid bilayer. It's going to alter the rate of transcription of mRNA on our cells. And that is going to bring about this increase in the metabolic rate. We'll talk about the examples of the different transcription factors or transcription processes that are influenced by thyroid hormone. Interestingly here, cretinism is a congenital disorder. This is what results if there is not enough thyroid hormone during development, during that fetal period. So if the mother is not taking in enough iodide, if she's not getting enough thyroid hormone produced, then the infant, when they're born, can present with behavioral challenges, learning challenges, physical impairment, and that is called cretinism, which speaks to the importance of thyroid hormone during the developmental period. So let's talk about the actual effects here of thyroid hormone. What does it do in the cell that leads us to say that it increases the metabolic rate? So it has a calorigenic effect, meaning that it pretty much speeds up the rate of everything in the cell. It increases the rate of the sodium-potassium pumps, okay? And kind of stepping back here and reviewing the sodium-potassium pumps function, that is what really resets the neuron after the action potential, right? So if we can speed up the rate at which the membrane potential can get back to -70, just as an example here, we can speed up the rate of the action potentials, right? So the speeding up of the sodium-potassium pump is one of the ways that thyroid hormone can speed up the metabolic rate of a cell. It also increases the number and size of mitochondria. We know that mitochondria are the powerhouses of the cell. This is where oxidative phosphorylation takes place. And so increasing the size and number of mitochondria increases the readily available ATP demand of those cells, which is another way that we speed up that metabolic rate. The third point here, speeding up or increasing oxidative phosphorylation enzymes, also ties into this increasing in the mitochondria, in the size, in the number, and that increasing providing energy demand. It enhances glycogenolysis, the breakdown of glycogen to provide glucose, proteolysis, the breakdown of proteins to release amino acids, and lipolysis, which is the breakdown of fats to release fatty acids and triglycerides. So basically just liberating energy stores to increase the amount of work that these cells can do and how fast they can do that work. It also promotes gluconeogenesis, which is the conversion of proteins into glucose. It's one of those back-end pathways that allows us to make glucose from proteins, as well as ketone synthesis, which is another kind of back-end pathway, which allows us to make ketone bodies from our fatty acids, right? Beta oxidation. And so it's another way of saying that the cells are looking for ways to increase their energy demand, to increase their energy production. And then the final factor here, it promotes the synthesis of beta adrenergic receptors. And we should all be making the connection here. Beta adrenergic receptors are going to be involved in the autonomic response. They're also going to be involved in the sympathetic branch of that response. And so that's the kind of effect we see with thyroid hormones. And a really good way to think about the overall action of thyroid hormone is to think about someone who has hyperthyroidism. They usually have rapid weight loss. They usually are quite anxious. They've got tremors, sometimes muscle spasms, just this overall heightened increase in metabolic rate of cells throughout the body. There's usually hair falling out, just this excess energy production, energy demand, and metabolic rate of the cell.